I'm Ai-Ming Bao from China, Zhejiang University School of Medicine. And I'm also from the National Human Brain Bank of China. So today my topic is the role of oxytocin in bipolar disorder. I will talk about our study from, in fact, postmortem human brain and to animal model and back. And we know that major depressive disorder and bipolar disorder are the two kinds of the most common mood disorders. And their pathogenesis is thought to be a multiple factorial. And we can see its gene, its development, especially early development when we were in our mom's uterus. For that 10 months development, it's very crucial moment for a human brain to develop. And also after birth, in the neonatal and childhood, we will experience a lot of events, and some of them are traumatic. For example, childhood abuse or neglect, etc. And then the environmental, especially the environmental stressors play important roles. When these roles are interacted, and some of us, our brain are geared to the higher gear, which means it's more vulnerable to stress. So our stress response system are more vulnerable to the stress or to pressure, while some of us are resilient. So that is the difference. That is what we thought for mood disorder versus so-called healthy control. And such a character will actually, for the rest of our life, the only thing that we thought how to deal with mood disorder is to live with them and try to reduce the stressor or the pressure in their life. Because the same pressure, the same stress could make difference in between depressed patient or healthy controls. But in fact, what is really happening is when these gene developmental or environmental factors are interact, they cause our neural network, which is the network that is regulate our mood, emotion, etc., in a vulnerable way, right? But what are they? They are mediated by neurotransmitters and neuromodulators. So that is what we call that very vulnerable neural network. To our attention is that the major depressive disorder, I can list the major or the core symptoms here. Usually among these 9 core symptoms, if one fit with 5 or above, then a major depressive disorder could be diagnosed. You can imagine that among these 5, nobody should be the same, which means even we are vulnerable to a mood disorder, the individual difference can be very significant. And bipolar disorder, we can even see from the development from when it's birth, it's very well, and then due to childhood to adolescent, the period can last quite long. And finally, when first, usually it's depression, and when the mania come out, then it's bipolar disorder. So, you know, these are all mood disorders, so it means they share similar symptoms, but also difference. That makes us to understand, therefore, for the pathogenesis, they have overlap. They should also have their distinct, their own character. And the necessity for postmortem human brain study is that we should understand that no animal model can very well mimic the full-blown of a human brain disorder. the development of the four. Yeah, usually it either mimic the genetic background or the early development or the stress from the environment. It couldn't be the mix or the interaction of the real human pathogenesis. And there are even no suitable animal model. They only can mimic symptoms of major depressive MDD or BD, right? They can't mimic suicide. They never get suicide. They are very resilient. And the animal model also needs to be validated, even if you found something interesting. And epigenetic factors are unknown for animal model, but we know they exist in the pathogenesis for human mood disorder. Therefore, for these pixels, we think that clinical study for DNA, for gene study, for MRI image study, and of course, animal model, very important, but human tissue hold the last word. Yeah. Therefore, we should also study postmortem human brain. So this is what my team, in collaboration with Netherlands Brain Bank, and the founder was Professor Dick Swab. We, in the past several, 10 or 20 years, we worked together to mainly study on postmortem human brain tissue. What we focus is on this HPA axis, hypothalamus pituitary adrenal axis, which is the key stress response system in our brain. You know, it also holds for the chronic process, which fit the most, more for human stress response. Because acute stress, it's usually neuronal, and we can say those neurotransmitters were taking over, right? But for a lifelong chronic disease, the HPA axis, which is originally help us to keep the homeostasis of our stress response, is hyperactive in depressed patient, also in bipolar disorder. But for this regulation, I will try to raise an example of the, I sometimes call it our fast speed train, yeah. So that is a map in our brain. It's a complicated network. For the individual difference, when Shanghai station has something wrong, it doesn't mean Beijing station is not affected, but also if you try to repair Shanghai station, you got effect. But if you go to another city, Hangzhou, or then you couldn't see the medicine effect in a quick way. For this idea, so why this is hyperactive? Who or which factors regulate this neural network hub, different hubs, right? Different key stations or even small stations. They cause this network unstable. So we published a series of paper. But today I try to focus on oxytocin. Why? Actually, the original inspiration comes from Turkey clinician, although I still don't know whom they are. But you see the publication from them. They measure the blood of bipolar patient and the MDD patient and the healthy control. And they found out that bipolar patient has higher plasma oxytocin level, and the mania episode are higher than depressive mode episode. But even during remission, this patient, bipolar patient, has a higher level than healthy control. So they raise this question, is the higher oxytocin a trait marker for bipolar disorder? And this is very interesting for us because we study neuroendocrine system. So we thought plasma oxytocin level do not necessarily reflect central changes. So we have to test the central change, right? Brain oxytocin level, because we know that oxytocin, besides its peripheral function, like the uterus contract during birth, etc., it has central effect. It is social hormone. It has, especially during acute period, it has anti-anxiety, anti-depression effect. But why it is, if it's too high, it is bipolar, it is mania, right? So it's very interesting to us. Therefore, we study postmortem human brain. You can see we matched MDD versus control, BD versus control. For such matching, I want to strengthen. It's not easy. It's not a 3 versus 3, 5 versus 5. No, no, no. It is as many as possible, but need to be matched for age, for gender, we call sex, postmortem delay, clock time of death, season of death, a lot of confounding factors. Yeah, antemortem medicine, etc. But we did it. For this technique, it is so-called quantitative immunohistochemistry and in situ hybridization. And remember, this is on postmortem paraffin embedded, formalin fixed, tissue. So it's not easy. Dr. Lei Guo, at that moment, is a PhD student, finished this job. And what she found is that in the hypothalamus, CRH expression, so it's in PVN. Yeah, by the way, we start with PVN because, you know, oxytocin is produced in PVN, paraventricular nucleus, and SON, supraoptic nucleus, and some accessory nucleus. These are the key nucleus that produce oxytocin and vasopressin and the CRH, etc. But PVN produce CRH, which is the central driving force for HPA axis, right? It is corticotropin releasing hormone. In the past, our study focused on CRH, and we found that it is increased, hyperactive in MDD and BD hypothalamus. So we also try to validate this. But besides this, we found out that for oxytocin, it only increases in BD, not in MDD. Yeah, this is interesting, either from protein level or from mRNA level. And we also study on postmortem human brain, the DLPFC, dorsolateral prefrontal cortex, and anterior cingulate cortex, ACC, this receptor expression. Why DLPFC? Because in the past, there are many studies showing that this area is playing a key role in this mood disorder. And we have this serious early publication already found DLPFC. It could be a target for BD treatment, like TMS, etc. The target, one of the key targets is DLPFC, right? So Dr. Qiyang Jie, during the PhD period, finished this analysis. He even finished proteomics. But here I try to show this qPCR study, frozen tissue. He found out the oxytocin receptor expression in BD, especially in male BD. So if you combine with the hypothalamus data, you will find out a very active oxytocin system in BD, especially male BD. And indeed, it distinguishes MDD and BD. Why? Because in MDD, we didn't find oxytocin system indexes. At that moment, my colleague, did this very nice tracing, virus tracing in mouse and published in Neuron. She found out either, so let's say both PVN and SON. So she brought SON at least from the mouse into our attention, that both not only have their central projection, peripheral projection, also central projection, while SON is always neglected in the past because indeed most of the central oxytocin effect were from PVN. But if SON plays a role, right? So we thought we should do it. So the young PhD, Tan Hong, she performed a chemogenetic study on the oxytocin-free mice, with the acute and with the chronic pattern of activation. Acute means just once chemical activation of either PVN or both. Why we also did oxytocin only in SON, and I will tell you the results. Why we do PVN plus SON? Because PVN has already been validated in human brain, and our major purpose is not for mouse SON, what a role it is playing. We focus on if PVN is already activated, right? Like a human brain in BD, then plus SON, what will happen? So therefore, this is our original design, but when the reviewer asked us to add pure SON, we did it, and we found some constant result. What she found is that if you chronic, because acute, you got a slight mania-like behavior. And now we understand that so-called anti-depression, anti-anxiety is actually mania-like, slight mania-like. Yeah, these two are so-called bipolar. They are opposite, in the behavior. But chronic mimic human mood disorder more, right? It is a long-term, everyday, last for 2 weeks. And then during the behavior test, also add activation via the CNO injection. She found that OD PVN, only PVN, you got depressive-like or anxiety-like behavior, especially in male mice, which means also in female, but for such a multiple comparison, multiple experiment, you have to do the correction, statistic correction, and for female mice, the significance lost. So the male showed this, and but very interesting is that although male show more significant change in behavior, but their MPFC in mouse medial prefrontal cortex mimic more human DLPFC, right? But to our surprise, the male didn't show significant change in MPFC, while female, although behavior not so much change, but their MPFC shows a lot of stress-related molecule change. So this is a sex difference. And for chronic activation of both, we found mania-like behavior. And at this moment, the MPFC also show sexual different change. So we call that activate hyperthalamic oxytocin led to a bipolar mood changes that is dosage dependent and also show sex difference. Actually, when we talk about the story as if one by one, but for the experiment, we immediately go to human brain for following the previous one. The previous one, we only study PVN. So therefore, we also, in postmortem human brain, study SON as well, right? Double nuclei. But for this moment, we also sort of another peptide, neuropeptide, that is almost like a twin to oxytocin, that is arginine vasopressin. We call it vasopressin, AVP. However, you know, for a long time, we see this interesting phenomenon. These two, peptide, they only have two amino acids different, but they show almost opposite effect in mood, especially in mood regulation. Oxytocin, usually, especially if acutely given, it is anti-anxiety, anti-depression-like. But vasopressin is the opposite. Of course, when it's depressed, you see higher vasopressin. So why is that? So we thought, okay, we also test vasopressin in the human brain. So this is the new match. You know, for a brain bank, such sample are increasing. Therefore, in the past, the matching strategy, which is also used for here, but some of the patients' brain material used out and some new entered, then you can validate sort of with independent sample. You validate what you found. Yeah, all the researchers are blind to each other study, right? So we matched such also MDD, BD, and healthy control with a sort of new cohort. And to put the long story short, because it was under revision, but just before I came, it is accepted. So, and even on PubMed already, so I can tell you the results. We also do the whole nuclear quantification. And we found out that, so this is a strategy for PVN, SON, yeah, and in between AN, accessory nucleus, but it is very difficult to separate from the SON. Therefore, this paper put SON plus AN, yeah, different from PVN. And found out again, so we are very happy that in PVN, indeed, BD, especially male BD, has a very high expression of oxytocin. But its vasopressin is stable. And in female, it is the opposite. It is the oxytocin stable, but AVP has a decrease. Here, there is a very short story that is the lithium treatment. Lithium treatment significantly increase vasopressin and even cause nephrology. So severe, then we have to exclude these confounders. And if you put them aside, you will see in female BD, you see a decrease of vasopressin. So what is it? This means the ratio. Yeah, the ratio of oxytocin to vasopressin is higher in BD, right? So this is the conclusion. In animal model, in mice, we see mood driven in the opposite direction, depending on the place and the duration. For the place, I want to say, if you only activate SON, although I must say, SON and PVN has mutual connection. That is true. But if you really focus on stimulate SON, you don't stimulate PVN. Not that you have two needles, you activate both. In that sense, if you only activate SON, no change, no change. And if you only activate PVN, you already see change. But if you you activate both, you see mania. That is what I mean by dosage dependent. It's not how much you inject. You from 1 milligram to 5 milligram. No, we mean different nuclei, the number of nuclei, yeah. Postmortem human brain said also this. So in animal and in human, we both found unusual activation of the oxytocin system. So if I sort of human patient, human, it is from genetic background, from early development, from later life. These group of patients, which are BD patients, they have an inborn oxytocin hyperactivity. This is my primary impression. I need more evidence to test. So now we go back to clinical blood measure. But is the blood can really reflect the brain? That is our next question, and we are now doing it. So with this, I want to say mood disorders characterized by changes in multiple stress-related molecules. So oxytocin, vasopressin, CRH, also dopamine is under study, also PCAP, etc. And with sex difference. This gives us nice reminder for future study, and especially for the postmortem human brain study. Thank you very much.